Okay, so welcome back to this next video in which we are discussing asthma and anti-asthmatic drugs. Okay, so we're currently in the process of discussing mast cell activation. Okay, and we've discussed that the best understood pathway of mast cell activation is the way that mast cells can be activated by the allergen in atopic asthma. Okay, so the allergen will bind to IgE molecules that are directed against the allergen, which are produced in atopic asthma and are loaded onto Fc epsilon R1 receptors on the surface of the mast cell. And what this then triggers is the cross-linking of Fc epsilon R1 receptors, because different Fc epsilon R1 receptors will have different IgE molecules bound to them, and these different IgE molecules will be directed against different epitopes of the allergen. Okay, and therefore you can get cross-linking of many Fc epsilon R1 receptors together. We've then discussed that these Fc epsilon R1 receptor cross-linked units relocate from normal plasma membrane to these special areas of plasma membrane called lipid rafts. And in lipid rafts you have this transmembrane protein known as the linker for the activation of T cells. Okay, now, in mechanisms that are still not particularly well understood, these cross-linked Fc epsilon R1 receptors can then activate non-receptor tyrosine kinase enzymes. Okay, so these are just free tyrosine kinase enzymes. Okay, they're just an enzyme capable of phosphorylating tyrosine residues within proteins. So I will show this here. Okay, so the Fc epsilon R1 receptors that are cross-linked together can activate these tyrosine kinases, and the main tyrosine kinases which they activate are what are known as lin kinase, okay, and also Syk kinase. Okay, now lin, I don't actually know what lin stands for, but Syk, I can tell you what Syk stands for. Syk stands for spleen tyrosine kinase. Okay, so the S is for spleen, the Y is then the single letter code for tyrosine, okay, and then the K is for kinase. So in single letter amino acid code, Y stands for tyrosine, hence why we abbreviate this down to SYK, not STK. Okay, right. So both of these enzymes are just free tyrosine kinase enzymes, which somehow are activated by the cross-linked Fc epsilon R1 receptors. And now they've been activated, what they're going to do is phosphorylate tyrosine residues on the intracellular aspect of the linker for the activation of T cells. Okay, so the linker for the activation of T cells will have lots of tyrosine residues on its intracellular side, which I'm going to show as Y, which again I'll remind you is the single letter code for tyrosine. And basically, these tyrosine kinases, lin kinase and Syk kinase, can now phosphorylate those tyrosines. So I'll repeat again that this step by which the Fc epsilon R1 receptors that are cross-linked together in the lipid raft to activate the tyrosine kinases is not well understood, but it's known that they do. Okay, so this leads to the activation of these, and now once these have been activated, we know what happens then. Okay, they put phosphate groups on these tyrosine residues. They convert them from being normal tyrosine residues to being phosphotyrosine residues. Excuse me. Okay, so tyrosine residues with phosphate groups attached to them are called phosphotyrosine residues, okay, and the activation of the lin and Syk kinases in these lipid raft regions nearby the linker for activation of T cells proteins is going to lead to the phosphorylation of the tyrosine residues on the intracellular aspect of this LAT protein and their conversion into phosphotyrosine residues. So these little purple blobs here, these represent the phosphate groups that have been added on to those tyrosine residues. Okay, now the process is just exactly like what would occur in a receptor tyrosine kinase. Okay, these phosphotyrosine residues can recruit all sorts of different proteins to the underside of the phospholipid bilayer and activate them and result in signalling cascades. Okay, now there are two signalling cascades that we are going to look at. These phosphotyrosine residues are actually capable of activating a huge number of different signalling cascades, but we're only going to look at two. One of them is going to lead to a calcium signal which causes the exocytosis of the histamine-containing granules, and the other is going to lead to the generation of cystenyl leukotrienes. Okay, so let's start off with the one that causes degranulation. Okay, so basically, this involves a protein within the family of phospholipase C gamma enzymes being recruited to the plasma membrane, okay, by these phosphotyrosine residues, then being activated and then breaking down PIP2 in the membrane to diacylglycerol IP3. Okay, so first thing I need to talk about then is the phospholipase C gamma enzymes. Okay, so PLC gamma enzymes. This stands for phospho, that's the P, and then lipase, that's the L, and then C, and then it's gamma. Now, phospholipase C gamma enzymes are a family of enzymes consisting of two members. Okay, so there's phospholipase C gamma 1 and phospholipase C gamma 2. Okay, and they are both phospholipase C enzymes in this gamma family. Now I want to stress, this is a very different family of phospholipase C enzymes to the ones that are activated by heterotrimeric G proteins of the GQ/11 type. Okay, so in that very famous pathway where you have a G protein coupled receptor activating an alpha Q/11 containing heterotrimeric G protein, the alpha Q/11 subunit with GTP bound goes off to activate a phospholipase C enzyme, but that phospholipase C enzyme is of the phospholipase C beta family of enzymes, not the phospholipase C gamma family of enzymes. The phospholipase C gamma family of enzymes are recruited to the plasma membrane by phosphotyrosine residues on the intracellular aspect of proteins like the linker for the activation of T cells. Okay, so let's show this then here. So I'll show my phospholipase C gamma enzyme, and I'm not going to say whether it's specifically phospholipase C gamma 1 or gamma 2, it could be either of them. Okay, and we'll have the catalytic site here, like so, and here's this binding site here for the phosphotyrosine residue. Okay, now, this binding site that the phospholipase C gamma enzyme has is an SH2 domain, okay, which is a type of domain that you find in many different types of proteins, which stands for Sark homology 2 domain. Okay, so this is just a protein domain that you find in a huge number of different proteins, okay, and the special properties that the SH2 domain has is that it's capable of binding to phosphotyrosine residues, and indeed the other pathway that we're going to see in a moment, which leads to the production of leukotrienes downstream of the LAT protein being phosphorylated, is also going to contain one of these SH2 domains. Okay, so this is our phospholipase C gamma enzyme here, so it's either phospholipase C gamma 1 or gamma 2. They both have SH2 domains, which mean that they can bind to phosphotyrosine residues on the intracellular aspect of our linker for the activation of T cells protein. Okay, however, that's not enough to activate the phospholipase C gamma enzyme. That brings it up to the underside of the phospholipid bilayer, which is extremely convenient because in order to actually work, it needs to be near its substrate. Now, what is the substrate for a phospholipase C enzyme? Well, it's phosphatidyl inositol 4,5-bisphosphate, which is present within the phospholipid bilayer. So you brought it up to be close by its substrate, basically. However, to activate phospholipase C gamma, you not only need to bring it up to the underside of the phospholipid bilayer, but you also need to phosphorylate it. Now, this probably is done by the Lin and Syk kinases as well, but that's a little bit contentious what actually phosphorylates the phospholipase C gamma. Okay, but to activate it, you do need to phosphorylate the phospholipase C gamma enzyme here. Okay, right. So you bring it up, you phosphorylate it, that then activates the phospholipase C gamma enzyme. Now what's it going to do? It's going to start breaking down a component of the phospholipid bilayer, which is for short called PIP2, okay? But the longer name for PIP2 is phosphatidyl inositol, okay? That's the PI, so the first P is for phosphatidyl, the I is then for inositol, okay? And then it's phosphatidyl inositol 4,5-bisphosphate, okay? So the P is then for phosphate, the second P, rather, and then the 2 means that you've got 2 of them. Okay, now, slightly more rigorously, you would abbreviate this down to phosphatidyl inositol brackets 4,5-P2, like so. Okay, so that you tell people which carbons of the inositol ring the phosphate groups are actually coming off, basically. Okay, so that's the slightly more rigorous abbreviation for phosphatidyl inositol 4,5-bisphosphate. Okay, so let's actually have a look at this molecule and see what phospholipase C gamma 2 is actually going to do to it. Okay, but I think I'll get a new piece of paper to do this. Okay, right, so we want to discuss the structure of phosphatidyl inositol 4,5-bisphosphate. Now we first need to take on that prefix phosphatidyl, okay? Now what that means is that the phosphatidyl inositol 4,5-bisphosphate molecule is going to be based on a molecule of phosphatidic acid. So we firstly need to discuss what is phosphatidic acid, and then we can extend it to phosphatidyl inositol 4,5-bisphosphate. So I'm going to draw this as though it's within the phospholipid bilayer, okay, which indeed it usually will be. Okay, right, so phosphatidic acid firstly. So I'll just draw the cartoon and then I'll explain the different portions of it. Okay, right. So we'll start with this horizontal line that I've now coloured in green. Okay, this represents a glycerol molecule. So basically, phosphatidic acid is the basis for a whole bunch of phospholipid molecules that are in the phospholipid bilayer. Okay, and it's based on a glycerol molecule, so it's one of these phospholipids that is based on the structure of glycerol, basically. Okay, now, in case you're not familiar with what glycerol actually is, its proper name is propane 1,2,3 trial. Okay, and although propane 1,2,3 trial is more of a mouthful than glycerol, it's a more useful name to us because it tells us exactly what we're dealing with here. It tells us that we're dealing with a 3-carbon molecule, that's the propane, where you have alcohol groups coming off the first, the second, and the third carbons of that molecule. Okay, right. Now, of those alcohol groups that come off the first, the second, and the third carbons of the glycerol molecule, you're going to attach things to create phosphatidic acid. Okay, so these 2 vertical lines that are here in orange, these are what are known as fatty acid molecules. Okay, and again, the proper name for a fatty acid molecule is a long-chain carboxylic acid molecule. Okay, and that tells us exactly what these are. They are carboxylic acid molecules, okay, which have extremely long tails, basically. Okay, those are very, very hydrophobic, and these are the lipid components of this phosphatidic acid molecule. These are what anchor this molecule in the phospholipid bilayer, okay, in this hydrophobic core portion of the phospholipid bilayer. Okay, so these are the lipid portions of the phosphatidic acid molecule. So fatty acids or long-chain carboxylic acids. And they've been esterified onto the alcohol groups that come off the first and the second carbon of the glycerol molecule. Okay, so off the alcohol group on the first carbon and the second carbon, you've had attached these very long-chain carboxylic acids called fatty acids. Okay, then off the third carbon's alcohol group here, this purple blob here, this represents a phosphate group, okay? And this is why phosphatidic acid is a phospholipid molecule, okay? Because in order to be a phospholipid molecule, the only 2 criteria that you need to meet are you have to have a lipid component and a phosphate group. So we've got a phosphate group here, and these 2 long-chain carboxylic acids that we've got esterified onto the alcohol groups of the first and second carbon, those fulfill the lipid requirement. So phosphatidic acid is itself a phospholipid molecule, and it is present to some degree in the phospholipid bilayer. Okay, however, the main components of the phospholipid bilayer are not pure phosphatidic acid itself, but the derivatives of phosphatidic acid. Okay, and the way that you get these derivatives of phosphatidic acid is by adding additional things onto this phosphate group here. Okay, now the derivative that we're interested in at the moment is this phosphatidyl inositol 4,5-bisphosphate molecule, PI45P2. Now this is a normal component of the phospholipid bilayer. It is a derivative of phosphatidic acid. We can tell that because it has the prefix phosphatidyl in its name. However, it's not an awfully common phospholipid to have in the phospholipid bilayer. Okay, there are other phospholipids that are far more common, and we'll see the main one in just a moment when we come on to leukotriene synthesis. Okay, right, so to convert phosphatidic acid into phosphatidyl inositol, what you have to do is attach onto this phosphate group here an inositol ring. Okay, now, inositol rings are 6-carbon rings with alcohol groups coming off all of the 6 carbons. Okay, so the proper name for inositol is cyclohexane. Okay, so a cycle of 6 carbons, and then it's cyclohexane 1, 2, 3, 4, 5, 6 hexol. Okay, so of every single one of the carbons of the cyclohexane ring, you have you have a alcohol group coming off, and that means that overall the entire molecule has these 6 alcohol groups, one off each of the carbons of the cyclohexane ring. Okay. In addition, every single one of those carbons in that cyclohexane ring has a hydrogen coming off. So note, there are no double bonds in this 6-carbon ring, they're all single bonds, which means that each carbon needs 2 more groups coming off it besides the carbons that it's got attached to it, and one of those groups is an alcohol group and one is a hydrogen. Okay, right, so that's phosphatidyl inositol. Now what we want to do is create phosphatidyl inositol 4,5-bisphosphate. So all we now need to do is add on 2 phosphate groups onto phosphatidyl inositol, and then we'll have phosphatidyl inositol 4,5-bisphosphate. But obviously they need to be added onto the 4th and the 5th carbons alcohol groups of this inositol ring. Okay, so let me tell you how we do this. Well, the carbon here that is attached to this phosphate group here, and by the way, it's the alcohol group that's attached onto the carbon that attaches onto this phosphate group. This carbon here is considered number 1, and then you just count round number 2, number 3, number 4, so we'll add on a phosphate group onto the alcohol group that comes off carbon number 4, and then onto carbon number 5's alcohol group we'll also add a phosphate group. And that will then create us phosphatidyl inositol 4,5-bisphosphate. So this is my cartoon for what phosphatidyl inositol 4,5-bisphosphate molecules look like. Okay, now, you might be wondering, well, why is that called phosphatidyl inositol 4,5-bisphosphate? Why is that not called phosphatidyl inositol 3,4-bisphosphate? Why is this carbon not considered the 3rd carbon? Okay, well, when you look at my picture, there obviously is no difference between the 3rd and the 5th carbon here. Okay, but that's how my cartoon is misleading. Okay, or rather, it's oversimplified, because there is a difference between the 3rd carbon and the 5th carbon, okay, but it's a subtle difference, okay, and it has to do with the fact that inositol does not just have one optical isomer, okay. Inositol actually has a huge number of different optical isomers, because if you can imagine, each one of these carbons off this inositol ring has an alcohol group and hydrogen coming off it. Okay, either the alcohol group can come out of the page towards us and the hydrogen can go into the page away from us, or it could be the other way round. Okay, and this gives rise to loads of optical isomers of inositol, because every single one of these carbons here has that same scenario. Okay, now fortunately in cells there is only one optical isomer of inositol that's actually used, and this is called myo-inositol. So if you ever hear anyone talking about myo-inositol, it doesn't refer to muscle inositol, but instead it refers to the optical isomer of inositol that's actually used in living systems. Okay, so there are 9 different optical isomers of inositol that are possible. Only one of them is actually used in living systems, and this is myo-inositol. And the carbons of myo-inositol are pre-named basically for us by chemists. Okay, it turns out that the carbon that attaches up to the phosphate group here of the phosphatidic acid molecule when you're creating phosphatidyl inositol is actually the first carbon of the inositol ring, and then you count round 2nd, 3rd, 4th, 5th, 6th. And it turns out that the 3rd carbon and the 5th carbon are not symmetrically identical. They are subtly different optically, basically. Okay, so phosphatidyl inositol 4,5-bisphosphate is a different molecule from phosphatidyl inositol 3,4-bisphosphate. Okay, right, so that's the substrate for all phospholipase C enzymes. Okay, so if I go back to my other picture, here is our phospholipase C gamma enzyme now recruited to the underside of the phospholipid bilayer because it's bound to this phosphotyrosine residue of the linker for the activation of T cells. It's now been phosphorylated, so it is active, and now what it's going to do is it's going to work on IP2 molecules in the phospholipid bilayer here. Okay, so if I draw this just here, okay, like so, let's colour in those different components, or at least try to colour in those different components. So here are the long-chain carboxylic acids there. Here in green, that's the glycerol molecule. Here in purple, those are the phosphate groups. And then in blue, we'll have the inositol ring there. Okay, now what's going to happen is the phospholipase C gamma enzyme, like all phospholipase C enzymes, is going to break down PIP2 by cutting that bond there between the phosphate group here that comes off the 1st carbon of the inositol ring's alcohol group and the glycerol molecule, basically cutting that bond between the alcohol group of the 3rd carbon of the glycerol molecule and the phosphate group here, basically. Okay, and that generates you 2 molecules. One of them is just a glycerol molecule with 2 long-chain carboxylic acids attached to it, which I'll show here. There is the glycerol molecule, and here are the 2 long-chain carboxylic acids. That molecule will remain in the phospholipid bilayer, and that molecule is called a diacylglycerol, and for short that's abbreviated down to DAG. Okay, so this stands for diacylglycerol. Okay, so that stays within the phospholipid bilayer after this cleavage. The other molecule, which was already dangling into the into the cytoplasm, that now falls into the cytoplasm altogether after the cleavage, and that is just an inositol molecule. So here's the inositol ring with 3 phosphate groups attached to it, coming off the 1st, the 4th, and the 5th carbons alcohol groups of the inositol ring. Okay, and that molecule is therefore called inositol 1,4,5-trisphosphate, and it's abbreviated down to IP3. Okay, so the I is for inositol, okay, and then the P is for phosphate, and because you've got 3 phosphates coming off the 1st, the 4th, and the 5th carbons alcohol groups of the inositol ring, it's called inositol IP3 rather. Okay, so inositol 1,4,5-trisphosphate. Okay, right, so those are the 2 products that you get. Okay, the one we're most interested in here is the IP3 rather than the diacylglycerol. Okay, IP3 now falls into the cytoplasm, and it's going to work on receptors that are in the ER membrane and cause the release of calcium from the ER. Okay, so let me discuss this on the next picture over here. Okay, so basically, calcium concentration in the cytoplasm is extremely low. Okay, Calcium concentration in the cytoplasm is usually around 100 nanomolar. That is extremely low when you compare that to the extracellular calcium concentration, which is around 1.5 millimolar. Okay, so extracellular calcium concentration is 1.5 millimolar, and I'll draw a cell membrane there, splitting them apart. That's a 15,000-fold gradient of calcium across the cell membrane. So intracellular calcium concentration is very low. However, you do have calcium stores inside the cell, okay, and one such store, or in fact the main such store, is what's known as the endoplasmic reticulum, okay, abbreviated usually just down to the ER. So this stands for endoplasmic reticulum. Okay, now, if you want calcium to go up in the cytoplasm of the cell, and this is a really important intracellular signal. It's probably the second most important intracellular signal after cyclic AMP. Okay, calcium going up in the cytoplasm can cause all sorts of things. The one we're going to be interested in is the degranulation of the histamine-containing granules of the mast cell. Okay, but if you want to generate a calcium signal in the cytoplasm, you can either let calcium come in from the extracellular fluid, or you can release it from the ER. Now, in our case, we're going to be interested in the release from the ER. Okay, basically, there are channels in the ER membrane, okay, which can be activated by IP3, and these are called IP3 receptors. Okay, which I'll abbreviate down to IP3R. So if we have our IP3 molecule here, here's the inositol ring with the 3 phosphate groups coming off the first, the 4th, and the 5th carbons there. So here is inositol, and here are phosphate groups. IP3 molecules can bind to the extracellular aspect, not to the extracellular aspect, to the cytoplasmic aspect of the IP3 receptor. Again, in fact, there are 4 binding sites for IP3 on the cytoplasmic aspect of the IP3 receptor. It's not thought that all 4 of them have to be occupied in order to open the IP3 receptor. 2 or 3 is thought to be the number. Okay, and the binding of IP3 to the IP3 receptor then triggers it to open, okay, and it's then a calcium channel which allows calcium to move from the inside of the ER into the cytoplasm, and that calcium can then cause loads of intracellular effects. Now, the effect that we are most interested in is that it causes the degranulation of these histamine-containing granules. Okay, so if I draw a picture of a mast cell again, here's our mast cell, here is its nucleus. Okay, remember the mast cell is full of these granules containing histamine. Okay, and when you get a calcium signal, what this is going to start causing is it's going to start causing the release of these histamine-containing granules, okay, and the histamine is therefore going to be released into the extracellular fluid, and that's one of the key commands that the mast cells gives to bronchial smooth muscle cells to tell them to contract, basically. And I realise I've just coloured my calcium in green, but never mind. Okay, so you're going to release histamine upon mast cell activation. That's why you get this calcium signal generated by the release of calcium from the ER due to the generation of IP3. Okay, right. So that's one of the signaling pathways downstream of these phosphotyrosine residues on the intracellular aspect of the linker for the activation of T cells. Okay, and that results in the release of histamine from the mast cells. In the next video, what we'll turn our attention to is another intracellular signaling pathway downstream of the phosphotyrosine residues of the linker for the activation of T cells, which results in the production of the cysteine leukotrienes by these mast cells.